Good morning. Good morning, everybody doing well this morning? Well, welcome to our final morning together for at least this National Patient Conference. I'd like to start the morning by thanking and recognizing our friends at Johnson & Johnson for providing our amazing breakfast this morning, so thank you very much. Our first session this morning explores how data is transforming our understanding and treatment of myasthenia gravis. The goal is to, of course, improve the care and patient outcomes across the entire MG community. Presenting today, we've had the pleasure of meeting, and I've had the pleasure of introducing Dr. Paul Strumph, who will be one of the presenters, and I'd like to add now to his resume the 2026 MGFA Ellsworth Award winner. Presenting with Dr. Strumph is Anna Richards, so let me tell you a little bit about Anna. Anna leads commercial at Vitaccess, where she partners with organizations like MGFA to advance and manage our patient registry. Her work spans global, digital, real-world evidential studies, ensuring the patient voice is not only captured, but meaningfully understood and applied to improve care. She holds a law degree from Toulouse and Bangor universities and a master's in translation from Cardiff University, and brings more than a decade of experience in translation and localization, including work as a project manager and legal and medical translator. Pretty impressive. So please join me in welcoming Dr. Paul Strumph and Anna Richards to the stage.
I'm wondering if we could have two mics so we don't stand at the podium. I'm sorry. Anyways, before the microphones get here, we're going to divide this presentation into two parts. The first part will be a description of what a registry does and how we moved from our current registry to the new registry, which is the Vitaccess registry. And then the last third of the presentation or so will be a presentation about what the registration process looks like. Thank you, Mike. Great. So if we can go to the next slide. You've heard welcome and introduction, so we can skip that one. As I mentioned up there, we'll talk about the evolution of the MGFA registry. Anna will provide information on the actual registration process of VRMG registry, and we'll pretty much share the transition from the first part to the introduction. Then you'll see the screenshots. Okay, thank you. And then you'll see the screenshots of what the process actually looks like. And then we will open for the second Q&A. There'll actually be two Q&As. I think it's important that people have their questions answered while they're fresh in your minds about why should I even consider a registry. So we'll go through those questions before we talk about walking through the registration process. I also wanted to remind people that right to the left of the big MG booth that's lit up is the Vitaccess booth. And if you are interested in finding out information about the registry, if you go up there at any time during the conference, leave your email, phone number, and they'll be in contact with you. This is not a commitment to join the registry. It's just permission for them to reach out to you after this. Did I leave anything out, Anna?
No, that's perfect. You don't need to leave your phone number, just email. So we're keeping it as simple as possible, and we'll send you over all the information about the registry so you can learn all about it at your own pace and see whether you'd like to join. You can even set up a call with myself or one of the team to answer your questions, so to really take you through step by step how this works. So if anyone feels they'd like extra support in this, whether it's tech or whether it's questions about it, we're here to help you.
Thank you. I'll go to the next slide and talk about evolution of the registry. So this is a collaboration between Vitaccess and MGFA. I'm not going to read the slide to you, but this partnership came about for a few reasons, which I will go into in the next slide. But essentially, MGFA wanted to turn over the running of the registry to organizations that specialize in running a registry. In the previous iterations of the registry, MGFA was a sponsor, like a pharmaceutical company was sponsoring a study, and then the registry was hosted by a group that hosts databases. And being a sponsor has many responsibilities, and instead of MGFA building up that internal headcount to execute one registry, we thought it was a better use of precious resources to work with companies that did that. And when I say companies, we've been through many iterations. First, we worked with a university. So that was not a company, but it was a university that ran many registries. Then that university did not want to move to the next level of what people expect from registries, the users, the experience, how easy or hard it is to use. And so we went and performed due diligence and had a bidding process, and then we moved to a company that became known as Alira Health. That actually was a company, a clinical research organization, and Alira Health was not in a position to fulfill all of our needs, especially the ability to, as I will explain, get additional information than just patient-reported information. And so then we performed due diligence again and found Vitaccess, our partner. This is a busy slide, but I wanted you to be aware in the first bullet that the registry that people have been involved with, and just can I get a show of hands of how many people ever registered for the MGFA PR, the registry that we moved on from? Okay, so I do see hands in the audience, and thank you very much. Your data still exists. Your data is being migrated over in a secure fashion from Alira Health to Vitaccess. That data will still be available for study. And those studies are academic studies by people like the physicians who have presented at today and yesterday's conference. And those studies are then presented at conferences and frequently are published in papers that are read by others in the field and therefore advance the knowledge about myasthenia gravis and also the patient experience in myasthenia gravis. So that registry is closed for new entrants, but that registry will still be analyzed. It's not dead, and your data has not been in vain. So now we move on to what the registry going forward will be, and that is the VRMG, the Vitaccess Real MG registry, and that is a registry that opened before MGFA finalized a partnership or sought the particular partnership with Vitaccess. So it is a successful registry in its infancy. This is not a company in its infancy. This company was the company that originated a registry that many people in the audience will be familiar with, My Real World MG registry. So this company has had experience in the myasthenia gravis field. And for people that were in My Real World MG, I know because I registered for it that it was much more easy to use than the MGFA PR in terms of entering information. So we've brought up the interaction page with patients to a higher level. And the other thing that is different, besides just the technology to make it more user-friendly, is the kind of data that's being accepted. The MGFA patient registry, MGFA PR, relied on patient-inputted data only, and one of the review issues when papers were submitted for publication and just in general was there was no documentation in a medical chart that the patient's antibody levels were what was reported, or that how long they had MG, or what type of MG they had, other than patient-reported data. And sometimes people get information wrong, and sometimes the questions aren't asked in a way that's easy to understand. And so the VRMG will be pulling data from electronic health records, your electronic health records, with your permission. And so that is something that we expect a lot of questions about later in the presentation, and we're more than happy to answer them. And the information that I want to let you know about now is really that the health data that is pulled is only the data that you give permission for, but it's also anonymized. There is no one at Vitaccess that has access to personal identifiable information that will link the information that is pulled with you as a person. So instead, when people are evaluating what is the clinical course of myasthenia gravis, they will look at a subset of people, like maybe it's people who live in North Carolina, or maybe it's males, maybe it's females, maybe it's people between the ages of 18 and 30. The ability to identify individual patients does not exist. And so that's something that should be very reassuring to you. So I did hit many of these points earlier, but this basically talks about why this registry is important. Your information is important because although everyone experiences MG in a very individual way, it's the aggregation of that data that allows investigators, specialists in the field, companies developing medications to treat myasthenia, to understand what the course of the disease is. Can the course be altered in a way that's beneficial to the patient? And so this does serve both the patients and evaluating outcomes over time. The MG treatment area is growing rapidly, and currently there is no central database in the United States where this information is kept. And it's our goal in partnering with Vitaccess that just like with the predecessor, MGFA PR, instead of people joining individual registries for maybe every therapy that you're on, that you might consider joining this registry so there is more data on more people, and that would not prevent you from, also joining another registry if you wanted to. So that's another important point I wanted to communicate. And it's also the safety of the new medications and the therapies that are being used in myasthenia gravis, which we need information about. And by entering the information from the health record, we'll have a very good idea of what are the risks of the various interventions for myasthenia gravis.
I'd just like to add to that as well. So I've been talking to a few of you about why is there a new registry? So aside from just the electronic medical records now being part of that and linked to the patient questionnaires responses, it's really trying to find the bigger picture. So you might find you do a clinical trial. That data will stay there in a silo, and it won't really be shared out and spread around. When you do these types of studies, it's looking at the real world. So it's actually seeing what happens beyond the clinical trial. What happens for people with comorbidities? What happens to people if situations change, if they become pregnant, for example, or all of these different scenarios where there's different subtypes, and it's really exploring the whole of this. So this is why there had to be an evolution for this to be the full picture. So we did find with My Real World MG And with the MGFA patient registry, we were asking a lot of people. We were asking a lot of questions and asking things that maybe you wouldn't remember. I don't know if I would remember when I changed a treatment for something. So this way, you don't have to. The EMR data is refreshed. Your clinician's doing it. You don't have to worry about that. It's pulled through, and it's all done on the back end of things. And you just have to answer a couple of questionnaires. It's really very simple. It's about 4 to 5 questionnaires per month. They take about maximum 15 minutes of your time, and you can get a gift card each month as a thank you for taking part. So we're really trying to create this bigger picture that isn't just data that happens in a silo. We don't really know what's happening to everyone across MG, and this is the way that we can find out.
Thank you. And maybe it would be great if you took that slide.
Yeah,
okay?
sure. So this registry is for all comers. Whether you've got congenital, seronegative, everyone is welcome in this registry, although at the moment it is for over 18s. So Elizabeth, I know you would be very interested in the research, and I really would like, well, we're hoping to have other studies that are run that explore as well the caregivers in this study and pediatric populations and beyond. We also have clinicians treating MG, so we have a number of clinicians, and a load of them, if you look over at that booth later on, you'll see Dr. Ali Habib, Dr. Kelly Gwathmey, Dr. Zabin. There's many clinicians who are involved in the scientific advisory board for this registry as well, and a number of community neurologists who are involved, and some of them may be your doctors who are already involved in this. They're able to also see, so they get data dashboards, again, completely anonymized, nothing would ever be tracked back to you, but anonymized data so they can help make decisions. Because we're really looking at, I mean, this big picture is, what are the treatments that are doing the best in the real world beyond the clinical trial? You know, what is affecting people who may have also diabetes? Is there a certain treatment that's working incredibly well for them, and maybe it's different for others? We won't know this until we have the data that can show this. So it's essentially working with all of you, working with clinicians, working with the researchers, we can find out these details. So we're essentially bringing everyone together, and it is a long-term registry. You can withdraw at any time, but we're aiming to go for 10 years, and that once we kind of reach to the end of the 10 years, we look to potentially renew and have another 10 years. We do have to give a certain limit. There are certain countries, so this is a global registry. It's also in the UK, Spain, Italy, and we're looking to expand Canada, Belgium. There's many other countries, Australia as well. So you do, for certain countries, I'm half French, it's very strict. You do have to say a defined limit, and then you can extend later on. So that's a little bit about how it's working. And for your side, you do have just the couple of questionnaires. All the electronic medical records will be pulled in through the back, and all of this will go into a data lake. The same as with the MGFA patient registry and Mara World MG, they're in a data lake. So researchers can do valuable publications on this, find out things that we've maybe missed, and find patterns that we may have missed in the data. And within this, you'll see there's questionnaires around your symptoms, treatments. There's certain ones particularly focused on maybe fatigue, healthcare use as well. And we are looking as well at when it affects your work and your productivity. So there may be new questionnaires that arrive in the future that get an even bigger picture of what happens, maybe as well how it impacts people who are caring for you as well.
So before we go to the next slide, where Anna will share the current results of the registry, I just wanted to recap from a patient perspective. I was a member of MGFA PR, and I am a member of this registry. The registry in the United States has about 20 sites, is that right?
Yeah.
And these are sites where your physician, where they're based, has agreed to be a site, and there's what's called an electronic case report form at that site that is filled out at that site for your visit. This registry is wonderful in that where I live, there is no site. So I give permission for them to access the electronic health records, and that electronic case report form, if you will, is filled out based on information in the health records. And so we're not only restricted to sites that are identified. So I think that's a great advantage. The other thing is, when Anna was talking about, you will be asked to fill out questionnaires. It's the ones you're familiar with, the MGFA, the ADL score, and one or two others. And the way it happens is you get a text message. In my case, I chose to be alerted by text. And then they ask, have things changed in the past week? And if I say yes, then a questionnaire gets sent to me, and it takes me very little time to fill out to get my gift certificate. And it's been a great experience.
And he rushes off to get his coffee.
Maybe you could introduce this.
Yeah, sure. So, as we say, it's still in its infancy, but we do have 176 participants already, and it's growing. We've got it across 27 US states. You might spot yours in that list. And we're looking at the age distribution of the participants as well. So this is very early level data that's coming out. And we do have a first publication that hopefully we'll be able to announce fairly soon, and we'll be able to communicate it through the registry as well, which will be the full protocol of the registry. So you'll be able to see step by step how this was built, everything that happens in step by step. So that'll be, hopefully I can announce that in the next month or so.
Thank you. So this does talk about recruitment. Did you want to go into that?
Yes. So as Paul was saying, it's quite interesting that you don't have to go to a site. So you don't have to go to the hospital or the clinic to be part of this study. You can join from the comfort of your couch. Sorry, British English. And it's a massive convenience. You don't have to go anywhere. And I know that is very important to people, and we've made it so that we've tried to be as simplified a journey as possible when you're completing it. I'll take you through in a little bit some slides that actually show you how the onboarding looks like, how you kind of register and join. And you'll see it's fairly simple. There are some elements we have to do. So we really need to make sure that it's you. We really need to make sure that it's going to be fully consented and safe, and you've understood what you're taking part in. So there are a couple of extra steps, but hopefully you'll agree that it's fairly straightforward. Yeah, sorry, go on.
Oh, I just wanted to bring attention to the box on the bottom right, the consent to contact. This is a great feature which already exists in this registry. So think about how you may have been contacted to gauge your interest in participating in studies that MGFA has sent to you. What happens is MGFA has a mailing list and will send out information and ask if this might be of interest to you. But it goes to people who might not even be eligible for the study. And we don't know, we shouldn't know who's answering yes, because if you remember these emails, they typically have a link where if you're interested, you click on the link. So MGFA, doesn't know, who is deciding to find out more information about this, because we shouldn't know, but it goes to everyone. So in this registry, because of the information that's there, they know if you're male or female, they know your age, they know what kind of MG, or the registry data lake knows what kind of MG you have. And so if an academic physician or a pharmaceutical company wants to do a study, they can say, we want females age 18 to 40 who are premenopausal, and then MGFA would send information, but would not know, who was responding back. So it would be a much more focused contact about potential studies, which probably will be a relief to you because you might not appreciate getting all these invitations to participate in studies you can't possibly participate in. So that to me is a great feature.
Yeah, thanks, Paul.
You want to take this?
Yeah, sure. So how can you get involved? So for today, if you just want to drop your name and your email, and you can come over to the booth. Mark's over there. Hey, Mark. He's waving over there. Then we can send you through all the information to get you started. If you wanted to go solo and go straight into it, you can follow the QR code and start the journey there. Once you're all set up, you can then start completing the monthly surveys, getting your gift card, getting your coffee, and you'll be part of the MG research community from that point. And I think really we're trying to convey to you really how simple this is. So hopefully you'll agree, those of you who are already in the registry and who are already participating. Some of you, I've heard that they find it quite fun to go through and complete that and then be like, yes, I've got my gift card. So yeah, and I think there's a sense of as well, you're being part of shaping the future of MG research. So everyone can be doing their part to the future of this. On the previous slide, we had really the break as well in the new therapies and existing therapies that people are on. This is where we can start to see what is working best. And I think it's going to be in the interests of a lot of you to know what is the most effective treatment pathway.
And I would like to just touch on consent, and then we'll break for your questions. You don't sign one line and consent for everything to be pulled from your medical record. There's a staged consent process, so you consent to certain things. And also you should know that the consent is not permanent. In fact, access to your electronic health records has to be renewed with your permission. And so that should also be reassuring. And I've been in the registry for more than 6 months, and at 6 months, I had to be reconsented and agree to continue to participate. So it's not going to be like subscribing to Netflix where they keep taking your money until you say no. Okay. There are some questions on the screen, but maybe we can go to the audience and see if there are questions that people would like to ask before we get to the screen. Or we can start with the screen. Oh, here's one.
This one over here.
Hi there, thank you. My question is more on the researcher side. So if somebody wants access to this data as a researcher, how do they get it? And is there a cost to doing that?
Okay, that's an excellent question. So registries, by nature, they're quite expensive to run. We have a lot of hospitals involved. There's payments that need to go to those hospitals. So on that side, if there's research that needs to be done on industry side, so of all the sponsors of this event, they can access for a fee. And again, it's fully anonymized, fully aggregated data. No one can identify anyone. We have a policy that for academic researchers, it's free. So we've always done this on our previous registries, and on this one, we've continued in the same way. And so for any academic researchers who are interested in doing any research on the data set, they are most welcome to come and contact us. Yeah.
And another avenue there, in addition to Vitaccess's policy, MGFA, as part of our research priorities, currently we fund, or will fund, as we have in the past, 4 proposals from academic researchers for $10,000 each. And that will go through a process at MGFA to make those awards. And then for that cost, it's essentially a pass-through cost for Vitaccess to do an analysis and provide tables, listings, and figures, or that would be part of an abstract or a manuscript. So there's more than one mechanism for the academic researchers to explore the registry. And I also wanted to bring up that with the pharmaceutical companies that desire assessment of the data in the registry, it's high level. It's outputs, it's tables, it's not, anything that could identify back to you. And just also wanted to let people know that this was the same process that has been performed in the MGFA patient registry back when it was at the university, when it was first started, and also when it was at the clinical research organization, Alira Health. So pharma access, or access to data by pharmaceutical companies, the way it's approved and the kind of analysis, that hasn't changed. And maybe we should be clear that when we talk about access to data, we're talking about analysis. They get the analysis data sets.
Yes.
It's not like they get a link into the data.
No, so they would never get raw data. They would never be able to look at all of the details, all of your history like that. There are some really specific research questions, and the data would always stay within Vitaccess data lake. So the data never leaves, it never goes anywhere. It's just the analysis and the outputs of that data, that's what's explored.
Thank you. Let's go to the screen.
Paul?
Yes.
Paul, sorry, just one question right over there.
Hi. I'm just wondering if once you have gone online, as I did, and filled out the information, providing my name and my email, will you actually get some sort of confirmation back in regard to what the next steps will be in the Vitaccess process?
Yes, certainly. So on the other side, we've got our study team who will contact you and reach out, and they'll organize, if you choose, so there'll be a link to schedule a call, if you would like to do that. If you'd like to go straight ahead, there's also the instructions for that. So it'll be a response in the email, and there will be the next steps of the onboarding, essentially. So either a call, or you can click on the link to start onboarding yourself.
Paul, over on your right side, your right.
Hi, just a quick question. You mentioned that for individuals whose physician is not one of the sites, one of the 27 sites, that they could still participate and that their data would be pulled through an electronic record. Does that require that their individual physician get involved and consent to provide the information? How does that work?
No, so electronic medical records belong to the person that they're related to. So someone would give their own consent for those electronic medical records. The doctor doesn't need to be involved at all. And we found that is actually one of the smoothest ways of having people participate who are very, very far from one of these sites.
Paul, let's go to the Slido, please.
Oh, sure. It disappeared, if it can come back up.
I saw that someone had asked about, will this be in Canada? So I was just going to add, yes, we are looking to expand the registry. So this does involve a lot of steps, so it can't be that we just suddenly open up in another country. There are many different compliance and ethics, so we will make sure that everything is compliant for Canada and the different provinces. We do have some differences in Canada that you have to go to different ethics boards in different provinces. So we did it with Mara World MG, and we hope to do it with this one as well.
Is Slido up, Paul?
Yes, yeah, it is. And so the next question is, who owns the registry data? It's Vitaccess. And what is being done to protect our health information that is being provided, especially for data breaches? I'll leave that to Anna, and I will try to find that slide. I'll be looking at the slides on my phone. I am not chatting with friends to find the right slide.
So, really good question. So, we've been working in rare diseases since 2017. The team is made up of very amazing specialists. We have ex-doctors, ex-pharmacists, but we also have compliance officers, tech ops team, and the work that we do is around patient registries, patient studies. So we are incredibly vigorous in what we do. Even logging onto my computer takes about 10 steps, and I am never able to see any of the patient data at a raw data level myself. We follow international standards in operations. They're called ISO standards. So we have them for data compliance. We have cybersecurity. We have dedicated IT teams that protect us. We understand data, it needs to be incredibly protected, and especially on this level. So we have systems that are FDA level protected. And so we can, and at the moment we've never been breached. And we hope that that continues, and we put a lot of effort into making sure that it doesn't happen.
We have 2 audience questions.
So the access to the electronic health records, like I have, several specialists within that, so will they have access to all of the records, or will it just be the neurology records?
So when it comes over, it would be access to all of your electronic medical records. It would be fully anonymized. No one would know that it's specific to you, because the aim of this research is to look, it will be anonymized, aggregated data. That would be the focus. You can consent, so if you have different insurance groups, you can consent to different ones, if that is a preference for you. But generally, this is purely consent-based. If you don't want to do this, you do not have to.
Okay, thank you.
Question to your left in the back.
Hi, I just wanted to commend you guys. This is very, very interesting. I think this is the future of precision myasthenia gravis. But I also had a question. Are you guys planning to add any other registry data points, like maybe digital biomarkers, et cetera, in the future?
We would definitely consider this. So it is a case of the more data, the better, because we can get a better and a clearer picture of what is happening. So as the registry evolves, hopefully this can expand. There are different elements to consider, and we would obviously there would be added consent levels. If people wanted to stay in the registry but didn't want to take part in taking a biosample, for example, they wouldn't be obliged to do that at all. So, but we are really looking to make this a very holistic, very complete picture of MG in all its aspects. And however, so really showing your experiences, your real life experiences, how is this affecting you? We have other sub-studies that can run off this registry, so it could be that you might want to take part in some interviews and go even deeper into how you're feeling. In just the case of a few days, people have been talking to me how they don't want to tell the doctor the full extent of how they're feeling. And I think there's things like this that they just aren't captured anywhere. So we really need to explore how can we be helping people. Could there be other alternatives than telling your clinician? What else can we consider? So there's a lot of aspects to this, and there's a lot of branch-offs of extra studies that can definitely be explored.
Paul, a question to your left.
Someone asked my question earlier, and then I just thought of another one. So that I do have different specialists, so I can list them out. But my question I thought of after she asked my question was, what about insurance companies? Because I've had the same insurance all through all the specialists. Am I able to do a consent so that you can have access to the insurance records? Like, would that help?
I don't know the answer to that.
I'm not as I don't think I fully understood the question. So,
I've had the same insurance company through all the specialists that I've seen. So would that help if I gave consent for you to have the insurance company's medical records, if that makes sense?
yeah, sorry, I was just thinking, so if you consented, because you've had the same one the whole time, they would actually be a lot quicker for you because you would just put the one provider in, and then that would bring in all the records from the one provider since the start. So it makes it a lot simpler for you, essentially, because some people might have to put multiple in.
Paul, we have time for a couple of Slido questions, if there are new ones.
Sure. Why doesn't MGFA own the registry data? So owning the data brings on responsibilities as a sponsor, and MGFA has decided that we want the sponsor to be a professional organization that's what they do, and whether it was a university or a clinical research organization or a company like Vitaccess, which in some ways you might think of as a clinical research organization but specializing in registries, we've decided that it's a better use of the funds that are provided to MGFA to have more efficient execution of the registry. If you think about it, the staff at Vitaccess, and they are administering how many registries at this time?
Oh, gosh, put me on the spot. We have about 16 concurrent studies going on at the same time.
So 16 registries and probably 15 or more full-time employees devoted to this. For MGFA to oversee the registry and perhaps administer the database would be a big expense. And also the Vitaccess registry got started before MGFA left the MGFA PR. So Vitaccess's train had left the station, so to speak. They had a registry model that, and we looked at a few of them, that really met our needs. And so we also didn't see why we should invest in something that was as good or better when that existed. And when you think about it also in terms of owning the data, Vitaccess and MGFA PR are using data that is provided to you through your informed consent. In either model, whoever owns the data has to be governed by an advisory board. And in the case of Vitaccess, there's a scientific advisory board that has many of the clinicians that have given talks here, but also that are known to the MGFA community. And for things to move forward and analyses to be done and policies to be implemented, the scientific advisory board, is giving, their assessment of that. So your data is safe, and it's almost, to me, less important who owns it, and with the ownership comes a lot of responsibility. And MGFA did own and does own the closed data set of MGFA PR. A lot of responsibility came with that, which we decided not to build ourselves out to compete with companies like Vitaccess.
Paul, we have time for one more Slido.
Okay, and what I'd like to suggest is that if the Slidos can be saved, we will somehow answer them and be able to communicate where the answers can be found. Let's go to, the Slido is not currently up.
Okay. Are there any more questions from the audience at this point? Any more questions? Okay. Oh, right here.
I was just going to say, just before Jenny, just quickly, if at the end of this session you've asked some of the questions, there's some really pertinent questions here. If you go over to where Mark is, where it says Help Advance MG Research at the end, we'll be able to answer all of those questions.
Hi, this will be quick, but as we end, could you just kind of quickly recap how all of this data translates into how people feel and function with MG? That'd be great.
You want me to take it first? And then you can clean up after I make a mess of it? Your question seems to relate to, let's just make it a patient-reported outcome, like MG-ADL. The MG-ADL is pushed out on basically a weekly basis, and if you answer that your symptoms have changed, then it asks you to fill it out again. It doesn't show you your previous responses because when the instrument was originally validated, people weren't able to look at their previous responses to see, how they should change it now. So the instrument has to be used in the way it was validated. But that data is collected, and so if you think about it, if a score, the higher the score, the worse your symptoms. So over a period of time, your symptoms will be mapped, with the x-axis being, the months, and the y-axis being the severity of your symptoms. And so that's just you, but now take 150, 1,000 of you who are in the registry. That information could correlate. So an investigator could say, I'd like to know, does the MG-ADL score, is it dependent on the season? Or for women, is it, the menstrual cycle? So these kinds of questions can be asked and then answered with the data. I hope I didn't oversimplify your question. Do I need to go a little further?
Can I just add like a really quick thing? So I don't like calling it data. It feels very impersonal. So I'm just going to share something super personal. It's a bit of a secret. I think my boss might have a little panic in a minute, but I'm actually pregnant. I'm in my first trimester, and and it's a time, you don't usually share at this time of the trimester. But I wanted to share with you because essentially it's a data point. It's a data point about me. It's just something that, would be codified in a data. But it doesn't share much of my real story because it just says woman pregnant. And I think really the goal of this and the aim of this is to really share not just the data, but what's going on behind. What are your experiences? And this is really a small part because a lot of it is your medical records. It's you completing some questionnaires. But this will evolve. There'll be more things. There'll be the interviews, as I said, if you'd like to participate in that research. There may be other aspects they might want to that we would like to explore. So the financial burden on people, the things where you can't do as much as you want to do, or you have to leave a job, for example. So it's really to try and create the full picture of what are people going through that are in this MG journey, and not just be data, actually have an output, actually see what is this in reality, what is MG and what does it mean to you? So this is what we're trying to achieve.
Thank you, Paul and Anna. Thank you so much.
We'll exit that way. Thank you. Congratulations.
Good morning. Good morning. How are we doing today? Oh, well, that kind of stunk. How are we doing today? Good. I know, it's a long few days. I know. Anna, congratulations. What amazing news. Congratulations. Just getting some high fives. Oh my goodness, our MGFA family is expanding. We're going to have more babies. I can't wait. Okay, so it is my distinct pleasure to bring up our next speaker. And I have to tell you, when I first started in 2020, I learned of him almost right away, but you have to understand, I couldn't even look him up because my predecessor, Nancy Law, whom some of you knew and I adored, she only referred to him as the Godfather. So, Mr. MG, that's another name that people call him. And so, our next speaker is actually Dr. James Howard, Chip Howard, he goes by Chip. And Chip is a professor of neurology, medicine, and allied health at the University of North Carolina, Chapel Hill School of Medicine. He's been a practicing neurologist for over 40 years, an author of 250 publications in MG. He's the director of the MG Clinical Trials and Translational Research Unit at UNC. He's a global lead investigator on several of the new approved therapies for MG, and a recognized leader in advancing MG research, clinical trials, and treatment innovation. And he has played a pivotal role in bringing new therapies from real world to patient care. And then on a personal note, he does beautiful glass blowing work, so that's something personal to know about him as well. So welcome, Chip.
Can we turn on this mic here? Thanks, Sam, and thank you all for the invitation to speak. I like to walk, so that's why I want to use this. I've been asked to talk about advances in myasthenic research and treatment. I can't cover everything, so I'm going to upset several of the sponsors here in the room. And I apologize for that. These are my disclosures. I consult for a number of companies. I help design the clinical trials that we're using in our MG space. And I want to say that we're in the midst of a revolution. And this revolution started in about 2011 when I was first approached to talk about a new therapeutic and was involved deeply in its design and led to an approval, and the space has literally exploded, and I'll show you that. But do we really need new treatments? I get this question all the time. If we look at what's happened to myasthenia in the 1800s, in which it was a very deadly disease due to respiratory failure, we made tremendous over 50, 70 years, first with antibiotics, then ventilatory assistance. Saved people, and the mortality rates plummeted. And then steroids came along in the late 60s, early 70s, and we made huge gains. And then plasma exchange, and we made even more gains in intravenous immunoglobulin, subcutaneous immunoglobulin. And you'll note by the 1970s, we were doing pretty darn well. And then we started adding new immune suppressants to our therapeutic regimen. And we chipped away and got a little further gains, but not tremendous changes like you see in the left side of that graph. And so do we really need new treatments? So let me ask you some questions. How many here in the audience take no medications for their MG? Raise your hands. How many in the audience take only one medication to treat their disease? How many take two? How many take three or more? This is why we need new treatments. You're dependent on multiple therapeutics to try and achieve some semblance of normalcy. So we've made these tremendous gains. But if you look at the right-hand side of the graph, I still think there are a lot of individuals with unmet needs. And those of you who are taking 2, 3, 4, 5 different therapeutics are part of that unmet need. There are subsets of our population that have other issues that contribute to this unmet need. And I think, in reality, patients with myasthenia have unmet needs, and we need to do a better job than what we're doing. And what are these unmet needs? Well, if you're on steroids, I will tell you that more than 80% of people have terrible adverse effects and hate them. It's a double-edged sword. It's like sabers that, in the Crusades that were sharp on both sides. On one side, it's very great. It's a phenomenal drug to treat MG, improve symptoms. But on the other side, we have side effects that we have to balance, and often they aren't nice. And so that's a problem. With our other therapeutics, about a third of the individuals have side effects that are not liked and are problematic. The other issue is that many of the drugs we use take too long to work, months, sometimes even a year before they even start working. And then it takes 1.5 to 3 years before we really see how well it can do. And that's a long time to wait. That's a lot of lost life, if you will. We have other better things we want to be doing in terms of hobbies, in terms of families, in terms of working, etc. And so that's a problem. And then we have logistic issues. If you are on plasma exchange, you've got to go to a specialized center to get plasma exchange. If you're on a periodic intravenously administered drug, you have to go to an infusion center, or in some individuals, particularly if you're not on Medicare or something, people can come into your home, but that's an invasion of your space, and many people don't like that. So there are these logistical issues that we have to deal with. And then we have individuals that the drug doesn't work. And if you look at all of the phase 2, early phase trials, and phase 3, what we call registrational or pivotal trials in myasthenia, as you know, the first part is what we call blinded. Half the people, sometimes different ratios, but half the people get the real drug, the other half don't. If you only look at those who get the real drug, 14 to 48% of the patients don't respond to that drug. And if we were smart and we said, well, this is a mechanism of action that should work, why aren't 100% of the people in that group responding? And they're not. Up to a half don't. And that's telling us something about the disease, and it's saying it's not a single disease. It's different diseases with multiple nuances. And we have to do better to try and figure out why they don't respond. And then we have other issues, and we look at what's the frustration with myasthenia. It impacts their ability to lead a full life. The thing that I hear most often is, I can't predict my life. The myasthenia dictates to me what I have to do. Each and every day is a little bit different. There's no normalcy. That's huge. And we have to do better to try and fix that. And then on the right-hand side are many of the things that we've already talked about. One of the striking things, and it was a shock when I first saw this data, is look at employment. So if you're in Japan, 27% of myasthenics are unemployed. About another 30+% have a reduction in income such that they can't take care of their family unit. That's terrible. In Australia, nearly 60% of myasthenics are unemployed. In Denmark, it's around 47%. In Thailand, 58%. Huge societal economic burdens that we're facing because we haven't gotten the disease under control. And these are the reasons why we're continuing to search for new therapeutics. No one is going to work in everybody, but we need to have a smorgasbord, if you will, that we can pick and choose. And our standards of what we expect of patients is changing. And all of it depends upon shared decision-making. You have to be an integral part of how you're going to be treated in each and every case. But our goal as clinicians is to make you disease-free with none or minimal side effects. That's our goal, not just to make you better. And when I started my career back in the early 70s, was that if I can get you out of bed, that was great. If I can get you out of the house, that's even better. I didn't care about side effects. We only had prednisone to give you. And so the ravages of prednisone took its toll. And then we got a little bit better. And then we said we want you to have minimal disease, but side effects were still an issue. And now we're pushing the bar even further. I want you disease-free, and I don't want you to have side effects. And that's the goal that we're coming to. So in 2011 started this revolution, this explosion, if you will. And it's demonstrable here when you look at the number of clinical trials in myasthenia by decade. And so, as you can see here, in the 2000s, we're down around 21 trials in that 10 years. And in 2010s, we were 81. Look where we are now. We're more than twice the number that we had in the 2010s. And we're only halfway through the decade. We're literally exploding. And these are the publications that have gone. And the last column only represents a half decade. And we've already exceeded the decade before. And so it's a logarithmic, it's a rocket ship going to the moon and beyond. And how have we done? Well, this cartoon sort of says it. Whoops. I guess the video doesn't play. I apologize. But he talks, if you remember this movie, and he talks to the guy in the cave, and he says, "You have chosen wisely." And we have chosen wisely. We have picked therapeutics that really work. And I'm going to show you some of the progress that we've made. I can't talk about it all. One, because of time. Two, because I'm not allowed to, because these are agents that are in conception right now and haven't even been announced that they're going to have a space in the MG arena, if you will. But the big question we face is, what do we target? This is our cartoon of our immune system. It's huge. Multiple interplays of our antibodies to our T cells and to our B cells, terms that you've heard, and to our cytokines and things like this. And which is the right one? Is there a right one? Is there more than one? And I suspect there are. But how do we pick and choose? And it's tough. And so we develop a concept based on a mechanism of action. I just spent the end of last week in a meeting with a company, and they have a novel idea of a therapeutic, a very unique way of working, and will it work? And so we look at how it works and what's its impact on the immune system and think, yeah, maybe it has a good chance or maybe it doesn't. And then here's some things we can do to explore that, and if it does work, then we can go further, take the next step and then the next step. But in the upper right, we're talking about antibodies and how do we deal with antibodies. And we've made progress in trying to remove antibodies from our system through the neonatal FC receptor. And we now have 3 therapeutics that are FDA approved to do this. And we can also know that antibodies activate our complement system. And we know that complement is a bad actor because it architecturally destroys that neuromuscular junction, which is a very uniquely designed system to allow nerve and muscle to communicate quickly and rapidly, and complement destroys that, which then impairs how nerves and muscles can communicate. And we've made progress there. And those were 2 asset classes that have paved the way in all of the advances that we've made. But look at the rest of the slide. Up in the upper left, we're looking at a variety of targets that are modulated by various interactions of T cells and B cells. And we'll talk about a couple of these. And in the lower right, we're starting to move in how do we remove long-term our antibody-producing cells with things like CAR T and long-acting B-cell depleting therapies. But we don't know what is the right target yet, and so we're still searching. And hopefully someday we'll find one that works for many of our patients, but I doubt if it will be the full thing. So where are we emerging? And we'll talk about some of these through immunoglobulin cleavers and complement inhibitors and FcRn and B-cell depleters, which is a big one that's under play now. And then we have a whole host of targets with interleukin-6, and then you may have heard about the April BAFF programs, and we now have 5 companies that are interested in this space based on some data, and I'll show you some data that has come out of China. And then we have other targets in terms of Bruton kinase inhibitors, and then there are many others that I can't even talk about. And we'll talk a little bit about CAR-T program and tolerance, which may be the holy grail, much like that cartoon that I showed you. And so antibody cleavers, these are drugs, and they haven't even come into clinical trial yet, but in experimental models, they take your antibody and they cut it in half, instantly, within seconds. And could we use this to treat rapidly evolving muscle weakness, myasthenic weakness, or myasthenic crisis? This is where I see it having a potential role. And these are going to come to trial in the next few years. But just imagine that if you develop a flu and your myasthenia starts to go haywire and you're starting to have some respiratory issues with shortness of breath or swallowing difficulties, to get an intravenous infusion and within seconds, boom, everything's gone and you're feeling back because we've just literally destroyed the antibody. We also have drugs that will actually promote the interaction of nerve-muscle communication. We call them CLC1 inhibitors, and they're under trial right now through a sponsor that sort of gives you an added oomph, if you will, when you take the drug. And it may be a replacement for mestinon, for pyridostigmine therapy, and not have those side effects. It's being tried in other diseases, and that those look very promising. The myasthenia trial is still underway, and I would encourage you, if you have the chance to participate, to get involved there. You're well aware of the complement inhibitors. This was the first drug class that gained approval. The first approval treatment for myasthenia after some 300 plus years of stealing from the transplant arena the drugs they use to work in MG. And antibodies, as they attach to the receptor to attack it, activate the complement system, which then goes down a pathway and it forms this ring-like pore, which we call the membrane attack complex, that literally drills a hole through your tissue, your muscle, leaks all its contents, and then the muscle fiber dies and destroys it. And by blocking that, we can gain clinical improvement. The first drug that we had was eculizumab. Back in 2013, it was approved in 2017, and then along comes its cousin, ravulizumab, which is eculizumab, and we've modified it, so instead of giving it to you every 2 weeks, we can give it to you every 8 weeks, and some of you may be receiving this drug, and it's administered by vein, intravenously. But one sees on the left-hand slide the blinded portion. In the top line in red are those who did not get the active drug, placebo, and in the blue are those who got the active drug. And you'll readily see that those on placebo get better too. And this is a problem we have in myasthenia, and this is why we do clinical trials. Does the drug really work? Or is it all just placebo effect? And we look for the difference between the two arms, if you will. And myasthenia is notorious for this placebo response. And it can be as high as 62% of the individuals who get the drug, who don't get the drug, get better. And you scoff and you say, how is that possible? And at Harvard, we're looking at an antacid back in the 60s, early 70s. The patients, this is a blinded trial. We're going to give acid to half of you and no antacid to the other half of you. And they told the people, you are getting the placebo, and they still got better. And so the psyche interacts with our immune system, interacts with many of our systems within our body. And this is one of the problems we face in trying to design and validate, does something truly work? And as you can see, the vertical line is where everybody gets the active drug, and those who are on placebo get even better. And then they parallel the other arm. And so its cousin ravulizumab shows the same thing. And here's the third complement inhibitor, zilucoplan. And this one's given subcutaneously under the skin on a daily basis. And each of these have their pros and cons. Many people, the business people, those who are traveling, like zilucoplan because you just pack your syringes in your suitcase and go travel. You don't have to keep them refrigerated, and you can keep these at room temperature for up to 3 months in Europe, a little bit less in the United States. But the pattern, they work very similarly, and as you can see, very rapid improvement over the course of 1 to 2 weeks. Around week 8, 9, 10, 12, they all seem to get the best, and then they all continue on. And so this asset class works rapidly. And the adverse event profiles, we don't have time to go into it, but I will tell you all of the new drugs that we've got approved to date have much better side effects than all the drugs that we use on a day-to-day basis before them. And then we have the neonatal FC receptor. The concept is not new. In fact, we knew about this back in the 50s. There was an individual named Sir Francis Bramwell who said infants born to moms develop their immune system from mom because mom's antibodies cross the placenta as a fetus, and they're protected when they're born, and then their antibodies go away. The infant develops their own immune system. We now recognize from some exquisite work by a woman named Sally Ward in the 90s and the early 2000s that this process is in every cell essentially, and in terms of in the vascular space. And this is why antibodies have a long half-life, much longer than many of our other proteins that circulate. And so antibodies are taken up into the cell, and they're bound to this FC receptor, if you will, and they're transported back to the surface and re-released into the circulation. And this process continues. That's why antibodies have half-lives of months. If they're not bound, they get shunted into what's called a lysosome, where they get destroyed, as you can see in the lower left on that right-hand graphic. All those little dots, those are antibodies that didn't get recirculated and got destroyed. And so along comes a drug, the first one out, efgartigimod, and we're able to demonstrate that patients got rapidly better, as fast as those on complement. And if we administered this here once a week for 4 weeks, they got better, and then their disease slowly came back because the drug was no longer around. The interesting story is that this whole process occurred in an elevator ride between two individuals, Sally Ward and a scientific officer, and they knew each other, and they were talking, and they said, "Oh, what are you working on now?" "I'm working on an FC receptor and blocking antibody clearance." And he said, "Oh, we have similar interest as well." And they ended up talking, and lo and behold, out comes our second approved drug class, if you will. We now have data that has been released within the last couple of months that not only does it work in those who have antibody to the acetylcholine receptor, and other companies have demonstrated that this asset class works in people with MuSK. We're now able to show that it works in those who have seronegative myasthenia. That's a huge plus, a true unmet need, and LRP4 also. It works a little bit differently. While the basic mechanism is the same, it takes a little bit longer to get there, as you can see. And so it's taking about 3 cycles to really see improvement, 3 cycles to see improvement, rather than 1 cycle that we showed with people who had antibody to the acetylcholine receptor. But it's a huge plus, and this data will be presented next week in Chicago at the Academy of Neurology meeting. So we have these drugs that work near where the action is. Complement at the neuromuscular junction, FcRn at the neuromuscular junction. But we're also thinking about drugs that work what we call upstream. So do we go high or do we go low? And when we go low, they work quickly. They have very narrow side effect profiles that we can demonstrate. But they have to be repeated. And that's why if you're on an FcRn or you're on a complement inhibitor, we have to continuously administer this to you to keep it going. So we're now looking at moving higher up in the immune system and targeting those. Slower to come on and work, but longer durability of response. Dosing intervals may be longer rather than every 2 weeks. We may be able to go every month, every 6 months, sometimes even longer than that. We don't know about its side effect profile because as we go up, is there greater side effect profile? We have to wait and see. And so one of the first drugs now approved is ineblizumab, which blocks B cell production, depletes B cells, and you can see that rather than improvement occurring within 1 to 2 weeks, that was really substantial. This is taking several weeks before we see 26, 32 weeks before we really see separation of the curves. Again, the top line's placebo, bottom line's real McCoy. But this is dosed every 6 months. And so we now have to balance the pros and cons of each based on your own individual needs as to what works best or what is best appropriate for you. And so there are other B-cell depleters that are being looked at. And the latest drug is one that's called telitacicept. This inhibits part of the immune system that signals the cell to make antibodies. It attacks it at 2 spots. One is called APRIL and one is called BAFF. This trial came out of China. It's now being replicated in the US, and that trial is currently going on. And there are 4 other companies who are interested in this same mechanism. And so we'll have to see. But one sees relatively rapid onset, not as fast as FcRn and complement. But the magnitude of response is huge, more than we've seen in our other trials. Is this unique to a Chinese population based on culture, based on genetics? We don't know, and that's why this is being repeated. This is in patients who have AChR myasthenia or MuSK, and this is given by subcutaneous injection once a week. So we have to wait and see how this is all going to play out, but very intriguing and very exciting. CAR T, I think, is going to become a new kid on the block, and there are 2 kinds of CAR T that we'll get into. One is based on DNA, and the other is based on RNA, which is the messenger that tells what DNA is to do. We have to harvest your cells. We do that through a process called leukapheresis. The cells are then taken, they're transformed in one by a virus and another through genetic manipulation, and then the cells are expanded and then given back to you. And each methodology has its pros and cons. There are trials with DNA CAR T. They're very small that are underway, but they demonstrate that improvement occurs fairly rapidly and then has been sustainable out. And the latest data is that it's a little bit longer than this. But it requires that we remove all of your lymphocytes prior. We have to give you chemotherapy because we have to make room for the new cells that are going to grow inside you when we give them back to you. And there's the potential risk of some modest to serious side effects. But it's promising. It has the potential of being one and done. Will it be a cure? I'm not going to use that term, but we could see long-lasting improvement. The other methodology is to look at your RNA, where rather than allowing the cells to grow and expand within you, we can obviate the need of chemotherapy, what we call lymphodepletion, etc., by growing this outside of you and then giving you a fixed dose. And there are trials underway with that as well. And so rather than growing the cells in you, we grow them all out in the test tube and then give them back to you. So we have to take your lymphocytes, leukapheresis you, but we don't have to give you the chemotherapy. And this is potentially an outpatient treatment. And so there are pros and cons to both. With DNA CAR T, it's potentially one and done. It's potentially very long, durable responses, but you can see the problems that are potential and don't occur in everybody. And much of this data is based on oncology, where this technique was first utilized, and it has very little application as yet in the autoimmune space, and I'm not sure we can translate one disease group to another. And it's quite possible that in the autoimmune space, all of this will be much less. The mRNA CAR T is that there's no lymphodepletion, it's outpatient, no adverse events of significance to date, but it's not one and done. It has to be repeated. And this is data that we've just published recently, where we're seeing durable responses out for a year. And the dark blue is placebo, the red is those who got active drug, and 6 one-week infusions, and then we simply follow people from thereafter. We can repeat this, and these are 2 of my patients. The first was an AChR positive patient, and one sees very rapid over the course of a couple of months. Her MG-ADL score, or this is the composite score, drops, and then slowly out around month 12, she gets worse, she gets retreated, and she's now out around 18, 19 months now, essentially in clinical remission. The second patient is one who is seronegative myasthenia. He too had a great response, and then around month 18 had worsening, got retreated. He's now at about month 14 right now, and he's starting to get a little bit weaker. Disease-free for long, durable responses. Is one better than the other? We just have to wait and see. The other kid on the block that's coming up is something we call tolerance. There are 2 companies in this space. One has just started an MG trial. The other will start their MG trials later this year, initially in Europe, and then we'll bring it. And here is the second company. It's a product called TOL2. This is animal data with myasthenia, and one by administering this compound, the disease goes away, not to return over the period of observation. And so we're resetting the immune system so that it doesn't recognize the neuromuscular junction as foreign, and that it can be rebuilt or reinitiated to be its own self, and the immune attack then stops. But we have lots of questions. Which hill do we climb? Do we stick with standard of care? Do we go with complement? Do we go with FcRn? Do we go with CAR T? Do we go with B-cell depletion? We don't know what is best in all of these. Is one better than the other? And this is where real-world data will come in registries like the one that was just talked about, Vitaccess, and another one called Gravis, are gathering longitudinal real-world data that will allow us to make these comparisons from drug to drug. And at some point, we'll get some answers. Clinical trials are an integral part of moving the bar forward. And we only do that because of you. And so I want to thank you for those of you who have participated in clinical trials. Giving of yourself is critical to allow us to make these advances. We must recognize that clinical trials have imbalances. They're designed for specific populations, and it's not the real world. But it gives us an idea of whether something works, and then the real-world data will allow us to really see how well it will come about. The problem we face is that we have no biomarker. We don't know in advance what kind of a drug you're going to respond to, and we desperately need this because we waste so much time trying one drug. Doesn't work. Let's try another drug. That could be a year. And if you cycle through 3 or 4 of these drugs, you've just lost 5 years of your life, and that's not acceptable. And so we desperately need this therapeutic biomarker. Our progress, though, has been sensational. And I don't use that term lightly. We've truly made phenomenal progress, but we have many unanswered questions. In fact, I don't think any clinical trial we do is worth it unless we pose another 100 questions to answer. So it's 1 step forward, 2 steps back, 3 steps forward is sort of an advancement. And it's your generosity, as I said, that has allowed us to do that. And thank you again. We can no longer rely on the U.S. to provide trial candidates because we have so many options available to us in our healthcare system, unlike many countries around the world. And this is a problem with health equity that we have to address. There are companies that have had complement inhibitors approved back in 2017 that still don't have them available for patients because they can't agree on cost. And this is a real problem that has to be addressed. And so our trials are moving to other geographic areas, to China, to India, to the Middle East, etc., where we're trying to get patients. This is where we recruit in the U.S. And as you can see, the darker you are, the more patients that have been recruited into clinical trials. And if you're light blue, it's 0. Huge areas of the country here in the U.S. and in Canada where no patient has been enrolled in a clinical trial. Not that there aren't patients, not that there aren't interested patients. It's that we don't have the ability to get you to trial sites. There's no MG expertise in your area. And so we have to rely on pharma to be able to get you to the closest clinical trial site if you're interested in participating. So make your voices heard. And with that, I'd like to thank you.
Chip, we have time for Q&A. Any questions from the audience? Any questions? We have a couple.
So I have a question about, so zilucoplan, which is a circularized peptide, can be used in combination with the antibody therapies. So do you have any experimentation with combining those types of therapies?
Yeah, so zilucoplan is a 14 amino acid cyclic peptide, totally different than our other complement inhibitors, which are full-size antibodies. So therapies that deplete antibodies cannot be combined with those that are full-size antibodies, like FcRn, like plasma exchange, et cetera, because you just suck it out. There is in case reports, there's small amounts of data looking at zilucoplan with these other compounds. There is a published study looking at zilucoplan with IVIG and plasma exchange, and you don't need extra dosing. You just continue your daily subcutaneous injections, and you maintain effect. With other depleting therapies, we don't have data yet.
Yes, hello. My question is, if you lower the autoantibodies using the FcRn systems, is the complement system still active and still continue to cause destruction at the postsynaptic receptor sites of the neuromuscular junction? And how do you deal with that? Or have they not looked into that yet?
Yeah, that's an absolute fascinating question. Thank you. I used to be of the belief that there is no treatment out that totally takes out your antibodies. You'd be dead. Therefore, you have continuous complement activation and attack to the neuromuscular junction. And I've been a longstanding advocate that you need to treat with both arms to be as effective. We now have some data that has come out that the antibody is different in different people. And the antibody actually can change over time in a given individual. So we're not completely sure that that holds true. Though I suspect that in some individuals, absolutely, you need to target both in order to be fully effective. Excuse me. But I'm not sure it's true for everybody.
Question to your left, Dr. Howard.
Have you seen cases of rapid B-cell repopulation after CD20 therapy? And do you think BAFF inhibitors could help sustain depletion in those patients?
Yeah, so there is no data yet of using individuals who've been on either CD20 or CD19 B-cell depletion who then had an inadequate response and then treated with BAFF-APRIL that I'm aware of. Though that's not to say it couldn't work, because the site of action is in different locations. But we have no data at this point in time.
Dr. Howard, are there questions on the Slido in front of you? How to control progress increasing symptoms of myasthenia, like drooping eyelid, extreme fatigability, and full body parts, and AChR antibody is positive. I think that becomes an individual question between you and your treating physician as to what options are available in terms of other therapeutic strategies, what is best in terms of your total healthcare picture, your comorbidities, et cetera, and in the US, unfortunately, your financial resources. But that's an individual decision, and there's no general rule. I'm a student, 17 years old, currently suffering from MG. What are the precautions for me for managing fatigability in daily life? Life as a student is pretty rough and active. I think one has to time activities to times when you're feeling strongest. I think it's worth discussing with your institution for modifications in terms of what can be done to help you. Can classes be on a certain floor? Can you have access to elevators? Can you have second sets of books that can be kept in the classroom and another set kept at home so that you're not having to lug these around? If you're having exacerbations of your disease that affect vision, can your testing be postponed or done in a different manner through a third party who's writing your responses, et cetera? It's those sorts of things. It's listening to your body to maximize your strength over time during these periods. If you're a woman, we know that in many individuals, menstruation will adversely affect myasthenic strength. And can we time certain activities based on that, be granted accordances, things of that sort, I think are important. Dr. Howard, 2 audience questions.
I'm just curious, what is trials or research for congenital is being done?
Yeah, unfortunately, the number of clinical trials for congenital myasthenia are less than what we see in acquired autoimmune myasthenia. And you bring up a good point, because When we hear the term congenital, we think about myasthenia that starts in infancy or before the age of 2, and that's incorrect. We've seen this syndrome start in adults in their 40s and 50s. And there's actually a movement to try and change the name to genetic myasthenia rather than congenital myasthenia. And the term myasthenia gravis, I think, needs to be altered just to myasthenia. But those are things that are happening. There are trials. There is a compound that is being looked at in genetic myasthenia that will stabilize the neuromuscular junction, but nowhere near the kind of interest, in part because it's so rare, in part many of the genetic myasthenics are in parts of the world where research is not done actively, Middle East primarily, it's a problem. We're not there yet. I don't have a better answer.
We have time for one more question.
Hi, Doctor. Great presentation. Thank you very much. Very informative. Traveling from Toronto, Canada, with appearance of a congenital myasthenic person, and just curious, have you had any experience with ephedrine? Ephedrine, our son, he takes ephedrine several times a day. It excites the synapses, and we have seen a phenomenal result. As a matter of fact, it's changed his life. Not all Alzheimer's fear 1, I understand that, but in his condition, congenital, it's really worked. And the second thing I just wanted to say, I just heard you mention a comment about separating myasthenia, congenital and separating. I can't wait for that to happen. I think that'll be a great thing for the overall concern, very much so. Thank you.
Yeah, ephedrine used to be a very common drug used in genetic myasthenia. In the U.S., it's gotten a bad rap because of the cardiac adverse events that are seen. So as a substitute, we're using albuterol. In Europe, it's salbuterol, which will do similar things, whether or not to the same degree. It depends upon who you read. But yes, it's been a common drug, and we've seen very great responses. Yep.
Dr. Howard, thank you so much. Thank you for
Michael.
your time today. Is she going up? We're going to welcome
Vice President of Research and Clinical Development here at MGFA. And I'm thrilled to be able to kick off this session on community insights and ongoing initiatives. We're going to be hearing today from Logan Hazellief from the Patient Advocate Foundation on community insights. And then later on, we're going to be hearing from Dr. Gülşen Öztosun from GW Medical Associates on 2 studies that are currently underway. So some quick background on our 2 speakers. So Dr. Öztosun, she earned her medical degree from Acıbadem University School of Medicine, and she's a physician by training and serves as a clinical research associate where her work supports MG clinical research. And Logan is the director of the Patient Advocate Foundation. The foundation is dedicated to helping patients navigate, access, and afford the care that they need. MGFA is honored to be a key alliance partner of the foundation in supporting those living with MG. And now I would like to welcome to the stage Logan.
Thank you so much for that kind introduction. My name is Logan, and I'm thrilled to be here with you all today on behalf of the PAN Foundation and Patient Advocate Foundation to spend a few minutes with you to share the resources that we have available for the MG community. Now, I have been with the PAN Foundation for nearly 2 years and with the Patient Access Foundation for just about a month. And don't worry, it wasn't that they sent the newbie up on the stage. It's actually because back in March, we announced a strategic merger between the PAN Foundation and Patient Advocate Foundation to create the nation's most comprehensive healthcare nonprofit dedicated to helping people navigate, access, and afford the medication that they need. In case you weren't familiar with either organization, that's okay. Both PAN and the Patient Advocate Foundation are charitable assistance programs that offer a variety of resources, including financial assistance, education, and patient advocacy initiatives for chronic, rare, and life-threatening disease communities, including MG. Together, ultimately, we're bringing together our resources for patients. With more than 50 that we are putting patients first, we're working to support comprehensive patient, including expanded access to financial assistance, ongoing free one-on-one personalized case management support, trusted education and engagement, research that have elevated to expanding our partnerships with organizations like MGFA, given the critical work that they do for the MG community and beyond. Before I can review 26, I do want to take a moment to share some resources that are available today for MG patients. We consistently hear from patients that medication costs are growing and they're getting harder and harder to access the treatment that you need to be able to live a regular life. And with that, a lot of people know they need that help, but they don't know where to find it. And luckily, that's where we can come in. Both the PAN Foundation and Patient Advocate Foundation have financial assistance programs available for MG that are active until July 1st of 2026. On the PAN side of the house, we have a medication copay fund that can provide you with up to $20,000 a year for your medication out-of-pocket costs, as well as a health insurance premium fund to help cover those premium costs. Patient Advocate Foundation has an MG fund available as well, which can help cover copay, coinsurance, and medication costs, office visit charges, as well as those health insurance premium costs as well. Eligibility requirements do vary from fund to fund at both PAN and PAF. However, there are some common similarities between our MG funds. First, you must be getting treatment for MG, and you must be receiving that treatment in the US or US territories. I will also note that US citizenship is not a requirement for this. We do ask that you're prescribed a medication or treatment that is on our list of covered medications. You also have to have health insurance that covers the medication or product you're looking to get assistance with. For PAN Foundation grants, it does have to be government-insured health coverage, either through Medicare, Medicaid, or TRICARE. But for Patient Advocate Foundation grants, any insurance type is accepted. We also have income level requirements, and specifically, it's that your income falls at or below 500% of the federal poverty level. And just to put that into perspective, for a household of 2, that's an income of $105,000, but for a household of 4, that's an income of $160,000. And if you're interested in applying for assistance or learning more about our available funds, I would highly encourage you to do so. The PAN Copay Fund is currently open and ready for patients to enroll. At either organization, you can apply online 24/7 at either panfoundation.org for the PAN Foundation grants or copays.org for Patient Advocate Foundation grants. Or on the other hand, if you're like me and sometimes you want to talk to a real person who can guide you through the entire process, we have dedicated staff available to do that as well. I can leave this slide up for just a second in case anyone wants to grab a photo. I know my camera roll is full of slides from this weekend. And like I mentioned, as charitable assistance foundations, we operate our funds based off of donations. And unfortunately, there are times when there aren't enough donations to support a fund and the fund will close. But we never want to be the last resort for patients. And that's why we created Fund Finder. It is a free resource that tracks more than 200 different patient assistance funds across 8 charitable organizations, just like PAN and PAF. You can follow any disease fund of interest, including MG, and sign up for notifications via email or text to be the first to be notified as soon as funding is available somewhere. You can visit fundfinder.org on any web browser or on your mobile device. In addition to Fund Finder, our National Finance Resource Directory is another great tool for the MG community and beyond. It's an online directory that can help you create lists of resources, both national and regional, that are dedicated to improving your access to care. The directory highlights a variety of resources, including discounted and free medical services, education and resource management, living expenses, and much more. Would really recommend you all visit patientadvocate.org to explore this resource. Now, hopefully that's enough build-up, and you might be a little curious as to why the PAN and PAF grants are only active until July 1st, and that's because on July 1st, we are launching Total Assist. It will be the nation's largest and most comprehensive patient's assistant portfolio that's designed to make it easier for patients to start and stay on treatment. At launch, Total Assist will include over 130 disease-specific and health equity funds. And in case you don't happen to have a pair of binoculars handy or a magnifying glass, I can confirm that Myasthenia gravis is on this list, as well as a category for non-disease support and services to help cover things like transportation to get to your appointments, which we know can be a big barrier for many folks, especially if you're in a rural community. We are firm believers that medication should be a relief and not a burden, and that's why Total Assist was built with you in mind. It's meant to be flexible, simple, and based off of what you need. With financial assistance through Total Assist, you can use your grant for medication copays, coinsurance and deductibles, health insurance premiums, office visit charges the day of treatment, and then administrative charges the day of treatment as well. And Total Assist does not limit how you can use these funds, meaning that you can mix and match your funding however you would like, that ideally to reduce any gaps in care. Ooh. I think I lost my slides. There we go, awesome. Like I mentioned, Total Assist is meant to be as simple and straightforward as possible for patients, which includes a simplified patient experience. We're reducing wait lists, including first time, first served eligibility criteria. You will find out instantly when you apply if you have been approved for a grant, so you don't have to wait and see if you'll be able to get assistance. We're also offering easy-to-use portals for patients, healthcare providers, pharmacies, and nonprofit organizations. And Total Assist will also include 6-month look-back coverage, which is going to make it easy for you to submit qualifying expenses that you incurred up to 6 months before you received your grant with PAF towards that grant cost. And Total Assist is just one example of an exciting initiative that the Unified Patient Advocate Foundation has on the horizon, and there are so much more to come. I would really encourage all of you to get involved and stay up to date on what is next. And the best way to do so would be to sign up via email or social media. You're welcome to use this QR code to learn more about our merger and Total Assist updates, hear about new programs and opportunities, or get involved with the PAF community. And of course, we are on all key social media channels and would definitely appreciate the follow. With that, I am happy to pass it over to all of you for any questions you might have for me.
Any questions from the audience?
No. It looks like there might be one on the Slido.
Great.
Does PAN or PAF help congenital myasthenia patients that take the same meds as MG? There aren't any that I have found. That's a great question. So within the PAN Foundation specific grant, I think it's the same across Patient Advocate Foundation, but would also just recommend checking. The covered subtypes for MG are congenital, generalized, ocular, transient neonatal, and juvenile MG. So really, as long as the medications are still on our list of covered medications, and if you have one of those approved subtypes, then you're eligible for assistance.
We have a question to your right.
Is the eligibility for Total Assist the same as it was for the current program?
That's a great question. So for Total Assist, specifically for our MG grants, it is government insured health coverage, that same income level requirements, and then everything else will be very similar. And if you are interested in learning more about Total Assist for MG or any other diseases, we actually have a webinar coming up on May 12th at 1 p.m. It's free. It's a 2-part series. If you're interested in registering, the URL is bit.ly/totalassist. And even if you know you can't make it or simply just don't have the time to go, I still recommend registering because we'll send you a recording of it after the fact that you can watch at your own convenience. That is going to go through all of the funds that we're offering via Total Assist, eligibility criteria, assistance amounts, anything of those details that might be helpful. The webinar date is May 12th. It's a Tuesday at 1 p.m. Yeah, the URL was bit.ly, so it's bit.ly/totalassist. Let's see if I can go back. But if you are able to sign up for those email updates from Patient Advocate Foundation, trust me, you'll get plenty of emails inviting you to that webinar and any more that are to come.
Logan, are there more questions on the Slido that you can see?
Not that I can see.
Any additional audience questions? No? Okay.
All
I
right.
think we're all set. Thank you so much.
Thank you all.
Thank you, Logan.
Hello again. It's great to see you all. Maybe many of you have attended the AI and digital health panel yesterday. I hope you enjoyed that. It was great to see your faces and then interact with you after the talk. But here I am to talk about 2 studies that we currently have at the George Washington University. So I'm going to talk about them a little bit. So let me wait for the slide. Okay, cannot see the slide yet. All right, here we are. Okay, so I will start with Bridge MG study. This one applies to all MG patients, and it's a survey study. It's completely anonymous. We really value, as researchers, to hear patient stories because, again, without the patients, all this research is really for nothing, so we really need to hear their stories, their experience, so that we can personalize the research and tailor it more based on the patient experience. So we have this video. I'll play it now.
We invite you to take part in the Bridge MG study, a nationwide survey for people living with Myasthenia Gravis. B stands for behavior, how daily habits and health behaviors affect MG. R is routines, how treatment schedules and lifestyle patterns shape patient life. I is impact, the real-life effects of MG on work, school, and relationships. D is for daily life, everyday challenges like fatigue, mobility, and independence. G is for goals, what patients hope for in treatment and care. E is for engagement, how patients connect with doctors, family, and support systems. This survey is anonymous, voluntary, and takes about 20 minutes. You can skip any question or stop at any time. Your voice matters. Join the Bridge MG study today and help shape the future of MG research and care.
All right. And yeah, your input really will help us understand as researchers to make care better. And of course, all the research that is being done right now should really have some patient voices. So the survey includes questions such as your life with MG. Everyone has their own unique experience, how it impacts their work, their social life. There are some questions about medicine adherence and relationship with your physician. And again, it's totally anonymous, and you can, I'll have a QR here. If you can have the time, you can take a picture of it. There are also some flyers over there at the MGFA booth. So yeah, I'll just put it there if anybody is interested in filling the survey. It takes around 20 minutes. You can come back to it anytime. and you can skip any questions that you think they do not apply to you. So yeah, okay. And this brings me to the second study, MuSK 1000. Anyone who has MuSK MG, this is for you. So what we do here is, again, just a bit of a background about MuSK. It's the rare of the rare. So muscle-specific kinase antibodies are found in around 8% of all MG patients. And again, predominantly seen in women, especially in their third decade of their lives. And there's a higher prevalence between African American women and also women of European descent. So there is some information that there might be a genetic risk factor or making patients more predisposed to having MuSK MG. And there are human leukocyte antigens. These are kind of like the safety badges that are found on the surface of your cells that tells your immune system, okay, is it self, is it not, is it enemy? So there's some genetic variations that also play a role in this expression. And again, a lot of MuSK patients mainly have bulbar symptoms, difficulty chewing, swallowing, speaking, so it can actually progress more severely. And that's why it's also important to understand if there are any underlying genetic risk factors. And our goal is to collect 1,000 saliva samples. And again, it's an ambitious goal in an already rare serotype of a rare disease, but we have close to 100 now. We started enrollment, so we're a bit behind on this goal. So we're really hoping to reach the 1,000. And of course, the saliva samples will be sent to the NIH to run genome-wide association study. So what is that? What is GWAS? It's really looking at the DNA and studying the DNA of individuals for both people who have the disease and for a control population who don't. So it's just sequencing the whole genome, the whole DNA of the individuals, and see if they're at a higher risk or predisposed to having the disorder so that we can understand it better. And usually we need a lot of samples to be able to do this. And there was another study, a GWAS study done for AChR-positive myasthenia gravis, but it hasn't been done for MuSK MG. That's why we're trying to understand that. So here's a Manhattan plot from that study. You see on the x-axis at the bottom, those are the chromosome numbers from 1 to 22, and then on the x, there is the significance, the p-value. All the little dots that you see, the red dots, are the kind of single variants that are more statistically significant, found in AChR-positive patients. And they did this study with 1,873 MG patients and a control group as well. So again, it matters because we need to learn more about MuSK. And then, of course, our research initiative to make this more personalized for patients, understand the disease better, and ultimately have a cure and even have biomarkers. And eligibility is, we need a lab report that proves that you have MuSK MG, and this is for patients residing in the United States as well. So the procedure goes, after we get your lab report, our research team reaches out to you, and we basically just ask some questions, have your consent, and collect some demographic information, and we send saliva kits to patients who are interested in participating. And then they mail it back to us. These are all prepaid, so we collect them and store it at our site at George Washington University to send it back to the NIH at the end of the study. And again, this is like a quick picture that shows how we do this. Okay. And of course, there's a unique ID that we assign to each of these samples so that they're not directly linked to your name and any of the information. And here's a study context. So if anybody as MuSK MG and would like to participate, please reach out to our study team, and you can directly email at musk1000@mfa.gw.edu, and there's a QR code to the website. So I'll also pause here if you'd like to take a picture and scan.
Thank you so much. Any questions from the audience? Any questions from the audience on these trials? One right over here.
Thanks for sharing those two studies. A question on the first one, the Bridge MG. What is your anticipated timing for sharing top-line results, and how do you plan to share those, whether that's as a poster, publication, like how's the data going to be disseminated?
Great question. We were thinking about this because, again, it's a survey and sometimes it takes time. I know that there's so many surveys out there and sometimes patients are overwhelmed. So we're really trying to end the survey by the end of this summer, August 2026. And of course, after that, we collect these responses on a secure platform called REDCap. So after the study closes, we will accumulate all this data, and then we will have a publication out of this to inform the medical community. So that's our plan for now.
Excuse me, could you verbally state the MuSK study email again, please?
Yes. So it's MuSK 1000 with numericals at mfa.gwu.edu. I guess I could go back. Well, the slide is not up. But was that clear? Okay.
Thank you so much.
Yeah.
We might have one or two questions on the Slido screen in front of you.
Oh, okay. I sent a saliva sample last year. How do I know it was you? But if you can email back to MuSK 1000, I can just check back on our database and see if it was received by the GW team.
Any other Slido questions? And from the audience, any additional questions from the audience? No? Great. Thank you so much.
Okay, yeah, thank you so much for your time.
Thank you. And let's welcome back Samantha Masterson.
All right. Hi again. I have to say how much I really appreciate each and every one of you, because we do understand travel and that some folks have to leave, and we do understand certainly that people are tired and may not be feeling well. But when we get to this last day on Tuesday, or even the end of this afternoon, we always have a certain amount of people with us still. And I just have to say thank you on behalf of everyone who has planned this conference. We really appreciate you sticking with us and being here for these final sessions. So just a warm thank you to each of you. So in order to put on a conference of this stature, and really anything that we do throughout the year, I always say a thank you for the folks who have helped support this conference. But the reality is that a lot of the people, the stakeholder groups that I'm going to thank, are people who are really very, very supportive throughout the entire year. So I just for a moment would like to thank these key stakeholder groups. I'd like to thank the National Patient Conference Steering Committee. These folks, as I shared with you the other day, spent many months planning this conference, being so thoughtful about the agenda, having ideas about the actual venue and the layout, and how we would have conversation, how we would format that conversation. And so I just really want to thank each of these individuals. They're not all here still, but if we could give them a round of applause, I sure would appreciate that. Thank you for your time. We could not have a program without the speakers, and so we really haven't spoken, and I own that because I've been up here a number of times. I haven't spoken enough about the speakers, but boy, are we just so eternally grateful to our speakers and the MG experts and those who want to stay close to the conference each year. We do switch up our speakers, but we have a lot of people who raise their hands and say they want to stay involved with this particular program. So it's a lot. They spend time with us. They plan their talks. That takes time. And then a lot of them move things around on their schedules to be, some of them flew in the day of their talk and flew back out, particularly if they were treating physicians. So just a real special thank you to the speakers. And thank you to our partners. Every year, our partners are just outstanding. I mean, really, program after program, as I shared yesterday, they're there with us throughout every year at everything, whether it's in the community at an MG Walk or a community health fair, or it's a larger program like this patient conference. They all followed us to the Netherlands last year for the international conference. They work with us on strategic partnerships, and just really, it's a huge support system for any patient advocacy organization. And as I also mentioned previously, they're a big part of the ecosystem. So just a huge round of applause for our partners. And we have MG ambassadors that represent the organization. And so if any of our ambassadors are still here sticking with us at this hour, could you please stand? Sure would appreciate it. So these ambassadors plug in different ways to the organization. They may represent, events in the community. They may represent fundraising. In the past, they've represented our registry and research. And so it's critical for us to always have the patient voice infused in everything that we do. I mean, that is why we exist. And so these are folks that give time throughout the entire year, and very close friends and support systems to the organization. So, and a lot of them are patients, people living with myasthenia gravis. So that speaks volumes right there about them giving of their valuable time. So thank you so much. Yeah, they deserve a second round of applause for sure. Our MGFA Board of Directors that work very closely with the organization, responsible for the fiduciary oversight of the business, and also lend a whole lot of guidance and expertise and strategic partnership on a regular basis. And as I shared, we had our Q1 board meeting here the day before the conference started. So it's always special to have that board meeting aligned with the conference, that they can all be here. So a special thanks to the Board of Directors. And we have two slides, the MGFA team. If the MGFA staff could please all stand, I sure would appreciate it. It'd be nice for you guys to see their faces. Most of them, I think, are over there. I could say so much about this team. It's equal parts, as much as we just care so much about our community, and we just appreciate all of you so much for the way that you support the organization. I would like to share that this team is really special. There's something very special about this team. I've been in patient advocacy for almost 30 years now, and I can tell you that you meet a lot of really good human beings. The great part about this work is that you get to work with the very best side of folks. You really do. The intention is pure. And so this is a team of professionals who have worked in this space for a number of years and who have really honed their craft. And we have assembled a really top-notch, high-caliber team that works day in and day out for this community. So I just have very, very deep respect and gratitude for this team. So thank you, MGFA professional staff. Okay, one more time, next year, the 2026 National Patient Conference. We hope to see each and every single person there next year and certainly tell other community members about it. It's going to be in Huntington Beach, California at the Hyatt Regency Huntington Beach Resort and Spa. We do have the dates. It's April 11th through the 13th. We will be sending e-blasts out. I see a lot of you with your phones. I'm going to keep this slide up for a bit. We're very excited, and as I shared before, we do try to do an East Coast, West Coast, so that we can really make it easier for people to come each year. Okay, no more cameras. Again, that will be our 75th birthday, so it's going to be bigger and better than ever, and we have some really, really special plans for that particular conference.
To see why our future is so bright, I think that I came up with this because I think our future is so bright for myasthenia as a whole, for the patient, the caregiver, the researcher, the industry partner, for everybody that's here. And I think we can see it through this program that we all put together here. And that all the speakers that were here, if we listened to them all and everything that they had to say, I think they brought it out that we can all see that our future is so bright in everything that they said. And I hope that everybody can feel that. And I think that that it was really brought out here that our future is bright. And I really hope that everybody felt that and that everybody can feel that our future really is bright for myasthenia gravis. And that, yeah, I really do believe it.
Thank you. Thank you very much. All right. Who else wants to share? Yeah. Mom and daughter, love it. Always in the front row.
Thanks, Sam. For me, the future is so bright because I've been coming to these conferences back when Nancy Law was, and every year it's the research is great to hear about. For me personally, though, every single year I meet a new person with MG, and that's last year I met Tim, and this year I met Paul. So every year I make a connection with a new person that has MG, and that for me is just mind-blowing because it is true what one of the speakers said, nobody really knows except another person with MG. So, thank you.
Also, MG has come a long way from the 1970s when my little baby girl, 6 years old, had it. I had to research on my own, and I printed out reams of papers, and I knew everything about MG. Now, I'm learning so much more.
Beautiful. Paul, let me go to Paul real quickly, and I'll come back. I know you didn't raise your hand, but she mentioned you, so here we go. Here we are. Was it your first conference?
I've been to 3 conferences now. Okay. I started in the one in Tampa, and then last year in Phoenix. So every year is a great experience because there's so much to learn, and every year has been really enjoyable, and I've learned a lot. So I just know the future is bright, and with what was shared today and this weekend about upcoming new drugs coming on the market, well, things are just going to get better from here.
Well said. Yeah, I'm going to work my way around the room. Just keep raising your hands.
Hi everyone, my name is Lucy. I'm here for the second time. I brought my mom. It's her first time here. My future is so bright because I'm so excited. I'm going to start a Spanish-speaking support group. I'm so excited. to reach out to my community where I'm in Phoenix area, and I just can't wait to get back home, start posting on Facebook, start getting the community, reaching out to all my friends to start spreading the word. Because when I got diagnosed at 14, I'm 51 now, my mom and my father were literally by themselves in the hospital, not knowing and what to do. They didn't know. We just kind of just went with on our own and trying to figure out what this life meant for their 14-year-old daughter. And so now it's so exciting to be part of you guys, to just, especially in the Hispanic community, it's, nobody knows. So when somebody, they feel alone. So I'm excited that we're going to be joining together, and I'm going to be learning so much from people who have done it already, and I'm just so excited. So thank you, guys.
Thank you, appreciate it. And we feel it. And there was 2 daughters in a row in singing their mother's praises, and I would just like to say hats off to the mamas and the papas, because I come from a rare disease family. I have younger twin brothers who are born with Angelman syndrome, and I've watched a single mom navigate the system when there is no information. So hats off to the caregivers. Did you want to say something?
I would say our future is so bright. One, I'm seeing more and more people diagnosed early. And 2, I know our mission is not to fundraise, and I've heard this before. We can't do our mission unless we fundraise. And I see this research and development, and through all of that, I see a cure. And isn't that what we all want? Yes, exactly, a cure.
Chip, do you want to say anything?
Well, I think our future is so bright because of the continued generosity of all of you who've pushed our research agendas forward. So thank you.
Thank you for your patience.
I was going to say our future is so bright, and a little bit Dr. Howard just touched on, is that a few years ago, probably I think maybe 3 years ago when I first started coming to this conference, a lot of like the rhetoric and the dialogue that I was hearing wasn't maybe, I guess MG is very diverse, and I'm noticing that the rhetoric and the dialogue with what encompasses MG is growing, and it's connecting much more with patients as we're moving along. And the other reason that I think that the future is so bright is because there's so much investment in us, all of the new research that's coming out, all of the new therapies that are being invested in, and all of that is raising awareness. And so for that reason, that's why I see the future is so bright. And in particular, thank you to Samantha and everyone here at the MGFA that has worked tirelessly to raise that awareness and to support us. Thank you.
Anybody? Yeah, okay. I'm just going to stay right here, work my way over.
Thank you so much. Well, I feel the future is so bright because, first of all, I'm a first-timer, but not a one-timer. I will be back. I will be back. Myself and my husband, we came from Canada for this, and I could so relate for my friend here from Baton Rouge. When this diagnosis came into our life back 40 years ago, we had nowhere to turn. We did all the work ourselves. We investigated, we studied, we spoke, we navigated, we networked, we did it all. And my only question is, where were you back then? But I'm so glad I found you because it's never too late. And the collaboration and the inspiration and the initiatives that are being put forth, make our future so bright. And I'm looking forward to the next chapter in our life, on our journey with our son. And I thank you so much. I thank you all so much for putting this event on and for welcoming us with such hospitality. I feel like I'm leaving with a whole toolbox of information that I can't wait to dig into, tap into, and network and speak to many of you that I have met. So thank you so much, and we'll see you next year.
Well, we're connected now, right? We're connected now. That's great. Leaving with a toolbox. That's what we want. Okay. Yes. Hi. Making my way over.
Thank you. We had a conference in Tampa. My question there was, how can we apply AI to MG? Guess what has happened in this conference? John Hopkins and GW. So they do listen to us in the conferences. Because after that question, about 3 gentlemen came to me. I don't remember them, but something happened. I'm very glad about that. So the future is bright. And before that, how many years ago, I asked Betty, my wife, I had to travel all night to New York City to the Today Show, 2012. It was the month of June. Yes, it was MG Awareness Month. And I wanted MG to be known by everybody. So I went early in the morning. 4 a.m. I was there standing online, and they asked me, they said, what? MG? What's this? MG what? They did not know what MG was. I had to explain. But look at it right now. Sitting in your living room, you see the commercials, right? On MG. So the future is really, really bright.
Really well said. Thank you so much. Thank you so much. And we do listen, right? We listen, right?
Yes.
We do. Okay, we're good listeners. Yeah. Hi, Todd. You didn't raise your hand. Todd's a new board member.
I mean, I think the future is bright because is there any other way to live life? Is with a positive attitude, and the placebo effect is real, as Dr. Howard pointed out. And so I think the only way to approach this is with positivity and with gratefulness, and happy for all of the movement that we have seen going forward with the treatments going forward. So I'm super proud to be engaged with this organization and look at it as a bright future as to where we're moving as a group going forward.
Who else? Why is our future so bright?
I was diagnosed 10 years ago. 10 years ago, the only treatment was Mestinon. And now, look at the long list of what we can choose from. In the next few years, there's going to be even more, and it's because of awareness, and it's because of the outcry for awareness and for people to understand that this disease is something that is out there. And this microphone is really heavy. My hand is trembling because it's heavy. I'm excited because in 10 years, look how far we've come. And in 10 years, look how far we'll be. Thank you.
Anyone? Yes.
Thank you. I just want to say that I have a wonderful family, but you now are my family. And I'll play the drunk uncle because of my MG. But this is a wonderful organization, and the future is bright because you're my family now.
All right, thank you. Lordy Lord, okay. Who else? Hi, guys. Our honeymooners. I understand it's the 4th honeymoon. How's it going? Yeah, I know it's going well. Okay, here we go.
Well, this is our 3rd MG conference, and the progress made since that first one in New Orleans has been highly significant, and it's already been mentioned. But a thank you to the Myasthenia Gravis board, the leadership, Sam, that you provided. It's a bright future, and adding new board members like you are is a significant part of that. So looking forward to Huntington Beach.
Okay, wifey has to add a few comments. And thank you, Sam, for your leadership. It's extraordinary. It really is. Yeah, you're amazing. The expansion of the support groups is amazing as well, and people should take advantage of the support groups, because if you haven't been to one, you don't know what's available. So check it out. The research, everybody's so friendly. I don't get it. What's going on here? Did you put something in the water? But the staff is amazing, helpful. I mean, it's just a brilliant conference and a way to bring us together. And your partners, the partners, they've been phenomenal. And they did all this extra work to provide the experience rooms. I mean, what else? Goodness, it was just great. Oh, and yeah, and the parrot, added a lot. I know, he was amazing.
You guys always bring the fun. Speaking of partners, hi, Lisa. Would you like to say a few words?
Yes. So this is my first conference, and I think what I've observed, I've been involved with the community for a little bit over a year and a half now, is the incredible sense of belonging that everybody, it's a culture that you don't experience much in many disease areas. So the culture here of community and belonging and inclusion is really an incredible thing to experience. So thank you all for helping me feel at home and welcoming me into the community with your open arms. And I echo everybody's sentiment about the bright future. I think from treatment to the expansion of programs and support, it's really an incredible space to be in. I think there's a tipping point moment happening in MG.
So when partners was mentioned, Lisa is with Argenic, so I just want you guys to know that's one of our great partners. Anybody else? Why is our future so bright? Hi, Mikey. How about a MGFA staff member?
Our future is bright for several different reasons. One, of course, the treatments and the research and the amazing groundbreaking discoveries that are taking place, laying a groundwork for our future. Our future is bright because of all of you in this room and MG patients and caregivers around the world, because it's so important to be able to take a look at yourselves and advocate for yourselves and speak up and have great relationships with your doctors and neurologists. So there's just so much progress and amazing things happening right now. This really is a trajectory that is growing and getting, going up and up and up. And we see it. There are tangible results every single day in the MG space. So that's really, really exciting. And then just finally, technology, innovation, all the amazing things that are happening. So those are the reasons why our future is so, so bright.
Thank you. And just so you know, if I walked up to somebody and they haven't raised their hand, and we all know how uncomfortable that is, it's because I know that Mike would add some value like that. And I know Lisa and I have great conversations about patient advocacy. So here we go. Another community member.
So as a first-timer here and fairly new to the diagnosis, I found out about the meeting and decided to come, but really having no idea what I would get from it or what I wanted from it. And I just want to say it was amazing. It was the kind of the best meeting I have ever been to, and I received so much that I didn't even expect of. Yes, I knew I'd make, new friends, and I did, and met my support leader in person and had wonderful conversations with so many people. I came into this feeling like I was alone on an island. I didn't know what was going on, and I came out of it feeling so energized and wanting to find how I can get more involved. And I just want to thank everyone for all the passion and the excitement that you brought, because it definitely, we feel it. So thank you.
Yes, I'll come to you after. Okay. Sorry. Here you go.
Oh, okay.
Did you raise your hand?
Yes, I did.
Okay.
But now, I think all of you pretty much covered what I was going to say. But I think our future is so bright because obviously of the advances in the medications and the clinical trials and everything. When I was diagnosed, there wasn't even an app for this or anything. And I mean, I have a lot of apps, but the most important thing is I have no social life due to this disease, but this is my second time coming to a conference. And if you see me at all, I've just been running up and down these hallways like nothing else. And the food, you guys, I mean, right? Like, how many times a day do they feed us? It's amazing. And I want to give a big shout out to all of you caregivers and my caregiver, my mom. I love her. She is my best friend. I don't think I would be here without her. Yeah. And I was talking to another ambassador, but I don't know if a person that doesn't have MG can be one, but she wants to get very, very involved. And literally, she's been out there to every tent, and they said that you had the most knowledge about this disease and so much more information than I've heard from anybody else. And it's true, because she does her research all night. She stays up doing everything for me. So I think if you don't have a family, or you say you don't have any friends, this is definitely your MG family. We are all on the same journey. Obviously, it might be different, but all we want is for a cure. And so that's why I think clinical trials are a big deal. I've already done 2. I'm going to do another one, because I don't want anybody else to have to go through what we're all going through. But yeah, and also I just want to say thank you to all of the sponsors and Dr. Vill and all the other doctors, Dr. Howard who presented, and you who is amazing, and everybody else. So.
And I want to say one thing. It was a choice between going to Nashville for a vacation or here. So guess what she picked? Yeah. This is where all her friends are.
Oh, thank you very much. Yes. And that's very sweet that yes, a spotlight absolutely has to be put on the caregivers. And Mama, if you want to become an ambassador, just let us know. Our team is ready and willing. Oh, that's funny. Yeah. I get the last word in our household, bruh, a lot. Yes. Wait. Sorry. I'll come back to you, okay? Promise. I had you waiting in the wings.
Okay, did you all hear it? Our future's so bright. Did you hear Dr. Howard say one and done today? So we're that much closer to a cure. And that's what we're all here for. We're all friends, we're all family, we're all going to stay together even after. But our future's bright. That cure's on the horizon.
Thank you, and hello everyone. My name is Candace. I'm here with Barb, and we are both caregivers to my dad, who was recently diagnosed in December. So this has been quite the journey from half a year ago, not even knowing that MG existed, to delving in. And I have to say, as a first-time attendee, I think I am feeling that the future is bright because of how many brilliant minds are working on this and how many dedicated people are working on this. If you're like me and you don't work in anything STEM and you haven't thought about biology since your intro to bio class in college, it is reassuring to know that there are so many people out there who are spending every waking moment of their day thinking about how to tackle this. And that's really inspiring. And I want to thank MGFA as well, because you're creating a dialogue for physician to physician, patient to patient, and seeing all of these different areas come together is unique, and it's not a given. So I want to thank for that. I want to thank you for that intentionality behind it, and for everyone who is thinking about this and working on this every day of your life.
Thank you. I just want to say something about your comment in particular. It is, I'll sit down. When you do this work, it's so true. And the team, it is such an honor and privilege. It's like we get a front row seat to the best of the best. So you're absolutely right. And I've never seen anything like it. And I've worked in liver and brain tumors and cancer. And the clinicians, the researchers, the labs that we partner with, to the industry partners, it's all so forward-facing and progressive. And I'll tell you, on the other side of that, there's just a lot of good humans. A lot of good humans. We all know that ego enters into everything in life, but there's a lot of good humans working in this space, doing a lot of good work. So, and then of course we get the utmost pleasure of working with all of you, the community, who make it extra special. And this community is very tight-knit. It's also a very well-versed community. You make it your business to understand what's going on, including the science, which is not an easy thing to do. So big round of applause for all of you. Everybody who's in the ecosystem, right? Anyone else? Oh, Pat, yes.
I'm going to try to say this without crying, but okay.
You don't have to rush. Just take your
Okay. The future is so bright because of all the technology, the research, the people, and the reason it's so bright for me is that this has allowed me to have my husband with me longer in my life. So that's why my future is so bright.
Beautiful. I'll give you a hug. That was beautiful. No. I'm not sure. If you put your hands up, put it down. Are we good? Okay. All right. Well, now I like this part better. I don't like goodbyes. Anybody who's close to me knows I don't like them. So the conference is wrapping up. It's not goodbye. We'll see many of you throughout the year at different community events and on our webinars. I hope to see each and every one of you next year in Huntington Beach. I want to acknowledge all of our virtual attendees. We had over 600 people. They're still hanging in and listening. This is the last session, so why don't we give them a big round of applause. And then just lastly, somebody had mentioned one of the last comments was about, and I'm so happy, it just fills my heart to hear what so many of you are walking away with. I thought the toolbox was a really cool comment, but let's not forget, connectivity is a health habit. We all need it. That's part of being human. So if we can play a small role in that, we're happy to play that role. So with that said, we do have lunch still. I know we fed you a lot. It was intentional. We want to keep you happy and awake. There's a wonderful lunch waiting for us outside. Take your time in the room and continue to have great conversation and socialize with each other. Thank you. Thank you so much.